Meta and Microsoft, as they share their requirements and how they're meeting the growing demand of data center performance. Of course, there'll also be networking opportunities, including a reception and a dinner to connect you with peers from across the industry. This summit is designed to be action-oriented and collaborative, with industry decision makers in attendance, helping you understand the roadmaps, the challenges, and the opportunities in scaling photonic-enabled cloud computing. And due to unprecedented demand, we've included exhibition space with all levels of sponsorship. But space is selling fast, so get in touch if you'd like to showcase your achievements for this influential audience. Don't miss your chance to register before the early bird deadline of September the 9th to save your spot and join us in Sunnyvale this October. We can't wait to see you there.
A brand new season of Optica online industry meetings start on Tuesday, August 26 at 10 a.m. Eastern, 3 p.m. UK and 4 p.m. in Central Europe. Join Optica's expert panel as we examine how optical imaging and visualization technologies are transforming healthcare. Powered by photonics and AI, these non-ionizing solutions deliver real-time high resolution for earlier disease detection and real-time surgical guidance. This ensures more accurate, minimally invasive procedures in oncology, cardiology, neurology, and beyond. We'll bring together surgeons, clinicians, medtech leaders, and photonic innovators to explore the latest advances in fields such as surgical visualization and endoscopy, laser-based diagnosis, fluorescence imaging, optical coherence tomography and multiphoton microscopy, spectroscopic and ultraviolet-enabled imaging modalities, AI-powered image analysis, and hyperspectral and photoacoustic imaging. And as usual, our meeting includes technology innovators, Brent Summer from Stryker, Frederic Leblanc from the Canadian Hospital Sears Shum, Austin Lindley from Copper Surgical Fertility Solutions, and Alistair Price from Siloton. And we will of course be joined by several technology enablers, Optica corporate members who can contribute solutions to integrate our needs, as well as share the latest milestones, several featuring our extensive coverage from Laser Wall of Photonics. The good news is that this meeting is free for you to attend thanks to the generous sponsorship of ANSYS. Enough from me, now let's start the live meeting. Thank you very much everyone. Thank you everyone for joining this spectacular session of the online industry meetings. I hope everyone had a fantastic summer holiday. It is great to feel all of you back. Today we are talking about medical imaging, but I wouldn't want to be fair without saying that I met many of you, most of you, during Laser Munich 2 months ago, and I could feel how good the business is going. This was a very difficult year for our industry. We knew it. We knew there were challenges, geopolitical challenges, exports, and it was difficult to innovate. And what we have seen, especially during Laser Munich and all the discussions that we had, is that all of you are doing a fantastic job. Summer was very useful to recharge, and we are all back. It is great to see great friends in the room, such as my great friends Antonio Raspa. Thank you very much for joining from Luxinar, the meeting today. Or my friend Stefan Weber from Faceform. You are doing a fantastic job. On Alistair from Siloton. Wow, you are eating the world alive. Siloton is doing such amazing thing these days. Welcome everyone. I had a great summer holidays. I'm full of energies. As you can see, let's start our online industry meeting on medical imaging. And I would like to say that today because of the support of ANSYS, we managed to bring together a really great meeting. It is very hard to get companies like Stryker or like Copper Surgical showcasing not what they do, but what they need from the photonic industry. Together with CRSHOM, the hospital in Montreal that already hosted an Optica online industry, an Optica summit, and Zeroton, a startup that is here to work with almost everyone in their supply chain to their presence. And with this, what we're gonna do is our usual game. We're gonna match the technology of those integrators with the enablers in the room with Epiphany, Chileas, Aquiris, Fabulence, Spaceform, ANSYS, and Flex Compute. Today is our first meeting on this final part of the season of our online industry meetings is medical imaging in 2 weeks. All of you active in quantum technologies, we have a great meeting on quantum key distribution, QKD, the tool that we need for the regular, safe, secure communications in this world. And I would like also to remind everyone that we do this because we love our corporate members. If you are not yet a corporate member, please join. If you don't know what the benefits are, ask Antonio Raspa from Luxina. He will tell you. We want our members to talk to each other and convince other members. It is great to have this growing community of members. Also remember as well that if you go now to optica.org/market reports, we have renewed completely the way to access the market reports. We have the largest database available in market reports, all of them for free for our corporate members. Optica.org/market reports. Make sure you download all of them. They are all for you. And also, I would like to say that as usual, we are live streaming this event in YouTube. So if you are in YouTube channel and you have any questions, write them in the chat and I will read them in the room. And also, I would like to remind everyone that you can watch this meeting as many times as you want because it will stay in YouTube for as long as YouTube exists. And now, let's start the meeting. And I would like to say to everyone that this meeting has been managed and put together by Dr. Helena Diaz, our Director of Laser Technologies at Optica. Helena, congratulations on working all summer. Helena, tell us what's going to happen on the next 1 hour and a half and introduce the first speaker first.
Okay, so thank you so much for coming. Thank you so much to all of our speakers. Thank you so much for all the people that is coming. It's coming from everywhere. It's coming from America. It's coming from Canada. It's coming from Europe. It's coming from UK. It's coming from Asia. It's really, really amazing to have this amount of people here. What we are going to do is what is going to happen basically in here. We are going to have a full conversation on what optics and photonics can do to help, to contribute, to improve the imaging during the medical procedures. So we are talking about surgery, we are talking about diagnostics, we are talking about basically how we can make a less invasive, more efficient way for all the medical procedures. Okay, we are going to talk also about how AI is going to contribute into the medical imaging. And this is a medical imaging in the sense that is non-radiative. So this is what we want to look for because we are looking for the less invasive procedures. Okay. We have system integrators like Stryker, SHUM Hospital, as Research Center, Cooper Surgical, and Syndeton. They are going to tell us what are the needs for their procedures and what they are doing. And we also have enablers in the room that they are going to tell what is the solution that they can apport on here. And they are looking forward for collaboration. Jose.
Time to start, and I would like to welcome to the floor our first speaker today, representing the company Stryker. It is an honor to have Brent Summer in the room. Brent, the floor and the attention of everyone is yours.
Thank you so much. Good morning, good afternoon, and good evening. Let me share my slides here. It's an honor to be invited to speak. Can you all see my slides okay?
Crystal clear.
Thank you so much. Well, like I said, thank you for having me. I'm the Director of Upstream Marketing for the Endoscopy division of Stryker, which means I'm responsible for leading the product definition and roadmap for our visualization, breast, and urology portfolios. So I want to start here with a bit of a quiz for this group. You don't actually have to answer, but I want you to put yourself in the shoes of a surgeon or clinician and help to spot the lesion in this lung surgery. So pretty good white light image, you can see pretty clearly, but it's not until you turn on fluorescence that the location of that, excuse me, pathology becomes particularly clear in a number of different modes. And I think that this really drives home for us the importance and the capability of advanced visualization, specifically fluorescence. So Stryker has been in the surgical visualization and camera business for over 20 years. And I'd say for the first 10 of that, it was really focused on improving that white light image. What can we do for more lines of resolution, better contrast, better color reproduction? But really there was a deliberate pivot about 10 years ago to bringing more rich content to the user, to that surgeon, to allow them to see more than white light alone. And the tools that we were using and continue to use today are largely fluorescence imaging. And that we are now 3 generations of camera systems in, supporting those visualization modalities that allow identification of lesions like you saw in the video previously. The way that we think about this is it's not our goal to provide a litmus test or a clear go, no go for the clinician. We want to complement the clinician's judgment and add to the existing suite of tools that they have within their ability to make the decisions on behalf of their patients. And that could be their education, that could be what they're seeing with their eyes or they're palpating with their hands. It could be preoperative imaging, MRIs and CT scans. But when you take the molecule targeting fluorescence that some of these drugs that are being developed by various pharmaceutical companies, and you combine that with a camera system that has appropriate sensitivity and maybe even more importantly, appropriate workflow, you now have another tool that allows that clinician to provide better care for their patients. One of the things that is important for me, and what we hear quite often, is that there are many different ways that we can provide additional information through advanced visualization. You see those on the left. There are fluorescent dyes. There's a number of different autofluorescent modalities on certain anatomies, and even just advanced imaging processing in general. But what we consistently hear is that most of the surgeons care less about how it's being done and more about the way in which it's implemented that you see here on the right. The first piece that we get asked, and it's very logical, is around the body of clinical evidence. So it's oftentimes not enough to just have an indication approved by the regulatory bodies and also have a feasibility study. A lot of times they'll want to see outcomes data. They'll want to see that there have been randomized controlled studies to build not only on the safety of it, but also the effectiveness and prove that these modalities are actually providing differentiation from that standard of care. And the second piece is reliability. And I mean that not just in the sense of that technology needs to perform day in, day out through reprocessing and the rough handling environment of a hospital or a surgery center, but also the sensitivity and specificity. Well, it doesn't have to be 100%. When it's being added to that suite of judgment, it cannot be a distraction for the surgeon. It has to be something that is a reliable source of data that they can add without a lot of cognitive load. I'm going to skip down to cost because that honestly is oftentimes one of the things that stands between adoption of novel technology and utilization. Certainly in the US and absolutely in Europe and other parts of the world, adding several $100s per case to pay for additional drugs or disposables sometimes can stand as a barrier between the user and actually that technology. And furthermore, if the cost of acquisition from a capital side to buy the actual equipment or specialized equipment is necessary, that provides an additional burden. But the last thing that is probably maybe even and most important And the thing that I often have to remind myself and my engineering team is the importance of workflow. And I'll start thinking about that from an intraoperative perspective. So during the surgery, and for us and for myself, when we think about product development, our product is the center of the universe. We think that that is the whole world. But when you are standing in an OR, you quickly realize that maybe 95% of that surgeon's attention is where it should be. It's with the patient, it's with the surgical plan, and you're left with a very sliver, maybe 5% of their mind share that's going to not just your equipment, but all of the equipment in the room. And then when you think about your camera or whatever equipment, that's even a smaller subset of that. So it is super critical when you deploy a modality that it's something that is easy to use, easy to interpret, and doesn't stand in the way of the surgical workflow or the broader continuum of care. So I'll leave you with a couple of different ways that we are currently on the market right now representing these fluorescence modalities. With the click of a button, again, super easy workflow, allows that surgeon to identify different anatomies and with the help of certain drugs, identify pathologies as well. So I want to thank you for your interest and your engagement, and I look forward to a great meeting.
Thank you so much, Brent. So this is one of the things is nowadays like using a lot. So I would like to ask you, like, what are the limitations in the fluorophore multiplexing? Because
Are you asking the limitations?
yeah.
I think that there's a couple, right? One is a lot of times one of the challenges comes down to depth of penetration and diffusion of light. Certainly, what we see with near infrared is anywhere in the neighborhood of about a centimeter, and anything that's deeper than a centimeter starts to really become quite faint and diffused. So we get asked that all the time, every week. How can I see deeper? And I think I kind of explained to them that, well, light does diffuse, and I don't have the knowledge that this group does, but at the same time, there's always that ask to say, are there different wavelengths, perhaps in the shortwave IR range, a little bit higher on the wavelength frequency that would allow a better depth of penetration. The other challenge is interference with white light. So we have the luxury with the near infrared and shortwave IR that it's not stealing from color reproduction of white light images, but when you look at dyes that fluoresce in the visible spectrum, like fluorescein is a great example, it becomes challenging sometimes to effectively represent what is the fluorescence of that and then overlay it on top of a full color image. Because going back to the point that I made about ease of use, a lot of times surgeons don't want to see just a simple black and white kind of X-ray equivalent image. They want to continue to operate with a full color image and then have the region of interest superimposed on top. So if you have a dye that's fluorescing in the visible light, it's difficult to do the analysis, and I apologize for not having the full background on that, but it's difficult to actually do the real-time analysis so you can put that superimposed on top without distracting from the white light. So those are two challenges.
Yeah, so this is something that I could be also help with the providers of the light sources. So what it could be the ideal light source for the fluorescence and also for the sensing?
You're asking me or the?
Yeah, I'm asking you what it could be your ideal for the specification. So.
I think that in terms of specifying, specific wavelengths and other imaging agents, I think that, continuing to be able to manage output of heat and intensity of light in that near infrared range and even in the visible range is an important one. I think that, from an ability to output any frequency or have the appropriate filter, I mean, that's not something that we're typically struggling with. A lot of times we do have a feedback from surgeons saying, hey, I want to put more light into the body, which is allowing our cameras to be more sensitive. But we end up being limited by heat management and thermal management, not so much at the source of light source, but as it actually travels up the light guides. You've got the light source, and then you've got your light guide, and then it obviously comes to the endoscope, and then it has a juncture there, and it goes into the body. And each of those different junctures has losses. And so in some instances, we're limited by the amount of light that we can put into the body by making sure that this is a device that's comfortable to hold in the hand, and even at the tip of the scope, you're not risking any patient burn. So while I'm not necessarily in a position to recommend the specifics of parameters for the light source design, that's another element of something that's a challenge.
Yeah, so
Ultimately,
we
the
have
light source, Helena, I'm gonna make the meeting what it is. Let's start interacting with all the participants and introduce them to Brent. Why we have seen that the light source is key for specificity, but I think, and you said it as well, filters are key. Nicole Sombotti from Edmund Optics, welcome to the Optica Online Industry Meeting. How do you think the good selection of filters could help Brent with the specificity requirements that he has?
Let me see. This was the
No,
question. Nicole Zambati?
the question is for
Hi,
me
this
to
is a
Nicole.
question for me.
Yes, Nicole.
Well, I think that there's a lot of optical filters out there that help control light based on what colors you are looking at. But I think a lot of what customers like Stryker perhaps maybe overlook is that if you customize those filters, they can be even more powerful in your system. So if you have specific dyes or colors that you're looking at in your system, there's certain ways to customize those, and every nanometer counts really when you're doing that. And that's a combination of your fluorophores and your light source, which each have their own individual peaks. And so if you can optimize where those peaks overlap, you can make a really high brightness, high contrast image that is even better for your surgeons that are out there looking for something specific, because I think as the medical industry advances, they're probably going to be looking for smaller and smaller lesions, for example, to catch things earlier before they become problematic. So as the medical industry gets more specific and is looking at smaller things, I think it's going to be important to really maximize the quality of those images. And filters is a great way to do that.
Well, I think
Brent,
that's
Brent, could you challenge Nicole? Could you give her a challenge for the request from optical filters that you cannot find easily?
that's a tough one for me. I think that the feedback that you've provided there, Nicole, is very, very helpful. And I think that our optical team I think does probably work with vendors like you guys to optimize that. But obviously, myself being on the marketing side, I'm always asking for more, because ultimately, like, being able to get, as you all know, more light into an image allows for less noise and higher sensitivity. So I see also in the chat you did have that question around tissue damage. So obviously we're always measuring the output of light at the scope tip as one of our many safety tests that we'll do, and it has to stay below a certain threshold. I don't unfortunately know what that is, but our safety team does, and then also scope tip temperature. So those are kind of the two areas. And one other thing that this is actually helps you think about from a workflow perspective, maybe less about like a proper optical design, but if you rewind the clock, one of the never events that happen more frequently than you'd think in an OR are drape burns or fires, because surgeons are frequently switching out their endoscopes, and in doing so they disconnect the light cord, which is putting out a tremendous amount of light, and then they'll either set it on the patient because there's a limited number of places they can put it because they have to handle sterile etiquette, or they'll set it down on one of the sterile drapes. And if that light guide is pointing straight at the drape, it can burn a hole in the drape, it can burn the patient, or even in worst case scenarios, it can light a fire. So one of the simple, and this kind of ties back to reducing that cognitive load on the surgeon, we have a simple feature where as soon as that light is disconnected, it has a little sensor that detects it's disconnected and turns off the light at the source. So, and then as soon as you plug it back in, it pops back on. So that's something that we've done to aid patient safety, and it's been hugely appreciated. It sounds like it's such a simple thing where you're like, well, yeah, turn off the light when you're not using it. But when you have so much on your mind, a lot of times that'll slip the mind of the clinician. So making something simple like that has helped a lot.
Brent, when you bring a Stryker to a meeting like this one, you will get always a lot of questions. We have a lot of questions for you. The next one is coming from the Netherlands. There is a design house for photonic integrated circuits called Epiphany, represented by their CEO, Jor Epping. Jorn, great to see you again. What's on your mind?
Yes, thanks. Thanks a lot. So I was thinking of you have these handheld devices, right, and have struggling with losses. So how about integrating, for example, the laser closer to the end in the handheld thing, in your handheld device, and that also plugs into the question from Chilas, of Dimitri. So what power and wavelengths do you typically use, and what do you need?
Those are all great questions, so I'll kind of take them one at a time. From a integrating the laser into the camera, I don't think that there's an opposition to that in any fundamental design. When you think about small diameter endoscopes that are maybe used for visualizing the ureters or the kidneys, and even some of the larger diameter, you will see like the full package, electronics packages at the tip, and we'll call that like a chip on the tip design. For us, when we think about we use a 3-chip sensor, and then the external light guide, which provides, great white light and fluorescence imaging. The idea of putting the laser inside, I think what's kind of paused that in the past is you would still require a light guide unless you want to move the entire illumination package, so the white light LED illumination as well as the laser illumination into the camera. And we get that question actually quite often. I was talking to a pediatric surgeon last week, and he was asking, hey, why, I'm working on patients that are 1 kilogram, 1.5 kilograms, 2 kilograms, and I have, all these heavy cords. Why can't you include it all in the camera? And that, if we were able to miniaturize everything down, that would be tremendous, and leave just a simple, power cable. But I think that some of the challenges go back to weight and thermal management. Some of these surgeries are going for several hours, and even though the temperature of the camera would remain well below what safety limits are, nobody wants to hold a hand warmer during 3 hours of high stress situations. They want to have a cool, as much as possible, a cool handle, and also the weight really matters. I mean, we watch, pulling grams out of the enclosure of the camera, optimizing the machining on the housing, because again, if you pick it up at a demo table, you're like, okay, this feels fine, but some of these procedures can literally take 6-7 hours. And if you're holding something for 7 hours, having a few grams makes a difference. So I would say fundamentally, no opposition to including from, an architecture perspective, including illumination, or even, moving further towards a true wireless solution. But we have to always be thinking about the workflow and the comfort of the surgeon. From a wavelength perspective, we obviously are looking at our white light illumination, and then also for the near infrared today, we're in the 780 to 805, depending on which generation of product we're looking at. And that's the near infrared fluorescent wavelengths that we need for ICG, which is a very common fluorescent dye in surgical applications. And in terms of power output, I'd have to pause and get back to you on that one. I'm not sure on what that power output is, and also what those subsequent losses are if you measure from the source on the stand all the way through to the scope tip.
Oh my God, I think people are extremely happy with what you just said. Brent, stay with us because Jorn also brought a slide to this meeting. Jorn, this is a really great time for people to know what Epiphany is. Jorn? Yes.
Yes.
So you could share the slide for Brent and for everyone. We have about 100 people right now for everyone to see it.
Thank you very much. I just need to make it bigger and yes. So we are Epiphany. We are photonic designers, meaning that we work on.
We cannot yet see your slide. So bottom of the Zoom window, green arrow up, and then select the screen that you want to share with the universe.
Yes, I forgot to press the blue button. Thanks. So yes, so we designed the photonic integrated circuits. So it's your vision and our expertise, because we've been have about 40 years of cumulative experience in integrated photonics, right? And just what Brent said, right? So I was thinking of integrating multiple colors on a single chip, which is a very small chip. It's handheld, and it's actually really lightweight compared to conventional lasers, right? But it doesn't stop there, right? You can use these multicolor lasers. They also nicely share one output, for example, in this example of Brilliance, who uses this for AR, VR applications, right? But you can also think of using it for Raman imaging, right? Which could be a topic later, right? Where you really scan about certain resonances and wavelengths to make chemical selective imaging. Yeah? But all these lasers that were used in the past can be miniaturized, right? Same goes for OCT applications where you want to scan it, right? And we really have an expertise in the different technologies, right? Because what you are doing with optical imaging is far from the telecom standard. So what we do is that we help our customers to find their way in a complex design and supply chain of integrated photonics, which is very chaotic. Yeah? And there we help our customers with the planning, with the design. We help with fabrication and can also support our customers in finding the right test partners. Then thanks, but I'm open
Thank you
for
very
any
much, John.
questions.
Brent, are you looking at photonic integrated circuits?
Yeah, I think, again, for me, I work closely with the development team, and I think that some of you guys may know John Fengler and team. So a lot of times they're the ones that are translating that to say, okay, this is something that this package is allowing us to miniaturize. And for me, I'm like, that absolutely fits the needs of what we're trying to do here. So unfortunately, I probably can't speak necessarily to the guts of the camera and comment on exactly why certain decisions are made, but anything that we can do that is reducing the power output, managing thermal, allowing better throughput of light, and also miniaturizing, all of that is tremendous help to us, and ultimately weighs in on the value to the customer and the patient.
Brent, you also, when we were asked about wavelengths, you said you love white illumination. Basically, you love all the wavelengths, and that is great. One of the best companies that we have in Optica providing very narrow and very wide tunable lasers is Chilas, who is represented by their CTO, Dimitris. Great to see you again, Dimitris. You also had a question in the chat. What's on your mind? And also after, show us your slide.
Yeah, so thank you for a nice introduction. I will pop up my slides because we are making tunable lasers, so they hit nearly any wavelength you can imagine. And our slogan is also Chilas lasers tuned to your wavelength. There, my slide deck should be popping up. We started off in 2019 with very compact light sources on chip technology. Then we developed a driver, and now we've got a fully furbished tunable light source which is ready to use. And there's a little bit of technology behind this, so it can be very nicely integrated to handheld devices. And that's also where Jörn was opting for. That we can make nowadays these light sources very, very narrow. They can tune through the whole gain bandwidth of the amplifier section there using a smart filter circuit. And they've got very narrow line widths. Also for coherent tomography, you can do these kind of things there into that there. So we've got several sources there. They're the size of a card deck. And this flavor shows you a little bit about the wavelength ranges it can handle. So if, for example, for visible wavelength ranges, you've got some 10 nanometers of tuning range. So an 850, the near infrared, I heard something around 780 and 805 nanometers. So there we have to find the gain section there, but we might be able to get a very nice tunable light source there, which is very fast switching also as well there. So, and also the telecom bands are there, but I think there's less medical applications there. We've got several projects there to highlight a little bit on the medical aspect where we find our place there. There's a Propel project where we focus on cancer detection and also hemoglobin testing of blood samples. So this will be developed around 1 micron of wavelength, and there we bring things together. So yeah, the goal is there to make a handheld device for testing your blood levels and for all kind of biomedical sensing. We've got another customer, they're doing photoacoustic imaging there, and we're not good in pulse lasers, but we provide an alternative route for detecting the acoustical waves coming from this tissue there with very precise laser lines onto a microphone there and to digest the acoustical signals out of there. So I think there we also have to switch to various wavelengths very rapidly and scan a grid, an array of microphones in one go. And I think that's what Chilas is doing. And yeah, what I see from Brent that there's a lot of applications going on there, and I think that there we could pop into one of the instruments you're making there to hit, for example, a spectroscopic feature there, or to hit another line there where you want to have a fluorescence or anything else coming out of there. So I see a lot of opportunities for TLAS there.
Very cool. Thank you. Thank you for sharing that. That's really interesting. I'd love to learn more about the photoacoustic imaging. I'll have to do a little research on that. That sounds like an interesting technology.
Yeah, absolutely. Thank you. Thank you so much, Dimitri. Thank you so much, Brent. Thank you so much, John. Thank you so much to you all. But now is the time to move all the way to Canada. So we are going to call to the floor Frederic Leblanc. So he's involved in multiple organizations. He contributes at the CHM Hospital and the Polytechnic in Montreal. He's also collaborating with a lot of spin-off that they are coming. Is reveals all the guidance like Esclaro and Reveal. And let's see, what do you have for us? So the floor and the attention is for you, Frederic.
Thank you, Elena, and thank you, Jose, for the invitation. It's a pleasure to be here today. As you mentioned, Elena, I'm a professor at Polytechnic Montreal, researcher at CHM Medical Center, and I'm also need to disclose also I co-founded a company called Reveal Surgical and Reveal Life Science, basically. So I'm going to discuss some aspects relating to those institutions and affiliations today. So we've built in Montreal an ecosystem, basically that the goal is to support the development and clinical translation and take to a regulatory approval basically of medical devices. Here in Montreal, the ecosystem is composed of universities, medical centers, including the CHM, for example, and one of the companies that we founded is called Reveal Surgical. The one of the system that we're supporting, and I'm using that system today because it's the one that got the farthest along the path to commercialization, and I'm going to show later an example of how it's used. One of the systems that we've developed is a Raman spectroscopy system. It's a fiber optics probe that's used to characterize tissue, and it's got onboard AI models to detect different pathologies. As an example, brain cancer in adults, basically. So one of the applications that we've developed over the last several years, and I would say we've done over 250 patients in neurosurgery, is the tumor detection model for adults in meningioma, metastases, glioblastoma, oligodendroglioma, and astrocytomas, as well as pediatric tumors. So the goal for glioblastomas, for example, some of you may probably know this, but the point is that there's no current technology that can really help that can really be effective in tumor debulking in glioblastoma and other brain tumors. And also no technology is sensitive beyond what you see on an MRI, on contrast enhanced MRI. So this is an example of a surgery where the tool is used. I'll just cut the sound here. So it's a probe that the surgeon can simply point to a location, and it provides a prediction based on an AI, as I said before, that we developed. The workflow of that tool is seamless in the sense that you can use the device under full ambient lights. It's a point and shoot device. You'll see now that the probe is going to be used in this case for the first time. And when you just point the device where you want the prediction, and here you see that acquisition and Raman and prediction of the eye in 2-3 seconds, and then it says cancer, for example. In a current study that we're conducting right now, our pivotal trial that's starting in 2026, basically, we've demonstrated that for contrast enhancing tumor, this is sensitive at 100% in 15 patients so far, and we're working out right now the limits of detection of this device for, I would say, a region around the tumors that are not necessarily normal brain. So one aspect that we've developed that's very key for these technology advances and commercialization of this device was to make sure that we could pick up basically anything that contaminates the signal, for example, extravasated blood. So we have this sort of it's an ML model that sort of predicts whether there's contamination from blood, basically. And if there is, the surgeon can simply retake the measurement easily. So that's for that system. And this is, as I said before, we're planning a launch in 2028 for that system with pivotal studies starting in 2026. I'm running out of time, basically, but this is based on, I say, 10 years of research, the specific neuro product here. There's lots of publications, but the deployment of live AI and demonstration that it really works seamlessly with no disruption of the workflow of the surgeon has been demonstrated only a couple of months ago. So a lot of work that's done both by sort of the through this ecosystem. Lots of the work that's done with the companies basically is developing the application for pediatric brain tumor and epilepsy. We have done like about 50 patients in breast cancer. We've done 100 for prostate cancer. Lung cancer is very advanced, and right now we're developing an application for colorectal cancer as well as others. So in conclusion, basically, we're really trying to develop this technology, and it's really consistent with what Brent was saying before. We're really trying to ensure that whatever we do just integrates itself seamlessly into current workflows. And also, we always pinpoint the specific need. You know, we're not aiming at replacing something, we're aiming at improving something. Like, for example, we've adapted the technology for biofluid liquid biopsies. So in this case, one example is, say you have a lung lesion, and you go through X-ray, like low-dose CT, for example. You want to see if you could improve the workflow of going to biopsy and other stages of treating the patient, basically. Could you improve the specificity of low-dose CT with a very simple liquid biopsy test, similar for PSA testing in prostate? So we're sort of adapting these technologies also for liquid biopsies. All right, so thank you very much for listening. So I'm happy to answer any questions.
Thank you so much, Federico. So this is actually good, and I'm very happy to see like how much it had been advanced. So Federico, you are a very good partner for us, and I have a very question for you. So what are the challenges that remain in integrating the imaging into the robotic workflow for this?
So you mentioned like the challenges for imaging and robotic workflows, basically. Is that what you said?
Yeah.
Well, I think that one of the advances also that we've done, I would say earlier this year, was to make sure we could adapt very easily the fiber optics to any context that's needed. For example, developing a flexible sort of fiber optics probe that could fit into a bronchoscope, as an example. For example, also we have these cases where we do endonasal and transocular surgery. So there is a specific design for practicality that needs to be done. Like needs to be a bayonet style probe, for example. It's a bit similar for robotics, right? You need to adapt, to which robot that you want to use. We've done some work with the Da Vinci robot, for example, for Intuitive, as well as the Ion robot. So, but there are, the challenges are multiple, Elena, but key is make sure you can adapt easily in terms of form factor, shape, size, etc., so that you have an instrument that can collect data that's consistent with the machine learning and AI model that you developed in the past, say, within another context. That's really that's the strength of the approach, I think, right now is sort of a compatibility of model across systems.
Yeah, so absolutely. So of course I have to ask you the Optica question. We know what you can do for us, but tell us what others can do for you. Tell me 3 challenges.
What I can do for you? Well, so that one of the things I really enjoy is talking to people. So, we have over the years, we've met with these different projects, and in particular the one that I presented, we met multiple challenges, right? There's so many traps on the way to commercialization when we're not there yet, but I'm happy to talk about this and discuss, help people if I can, basically.
Yeah, so, but what are the towards commercialization, but right now, what is your major challenge?
Major challenge basically is to take these modalities to imaging. So we've made some progress on that front, but specifically we need Well, there are things out there that we haven't tested yet, but we need very high dynamical range and high sensitivity cameras, basically, to make sure we can capture in imaging mode this sort of hyperspectral signature that's needed to do what we're doing with these single point probes, and also to make sure to see if we can perhaps reduce the amount of data we need to make the same prediction, helping basically getting to a point where we can do real fast imaging. But this is research, and this is really where we need help from the community and the Optica.
Helena, what a great
We
way,
have-
Helena, to start the meeting with a hospital, with a striker. Frederick, great to see you again. Let's go to
Thank you.
Boston, very close to Montreal. Let's go to Boston. Vivek, you have a question for Frederick. Vivek from MIT.
Yeah, thanks. First of all, thank you so much for this great talk. I have a quick question. You have shown the is reaching 90% while you are doing imaging. But what is the difference between the true positive and what are the overlap between true positive with the true negative, you know?
Yeah, okay. So you know that like true positive, true negative, sensitivity, specificity, you can quote anything you want. It's like, you know, overfitting data with the machine learning model. So, but the thing is that the context in which I'm quoting these numbers today, I can say, for example, that the sensitivity and specificity of tumor detection in brain, for example, when the tumor was showing contrast on MRI, is up higher than 95%. And when we're sure that it's normal brain, you know, it's same basically. However, you know, depending on the type of tumors that you're looking at, like for gliomas, for example, they're invasive. So what is the extent to which you can make predictions in the in-between region is something we're working on right now, basically. And this is where sensitivity, specificity will mean really something in the sense that how good are you at these ambiguous areas where oftentimes there does not even exist an actual histopathologic gold standard to detect what you want to detect with the optical system. So I'll stop there, but there's a lot to discuss around those points.
Yeah,
basically.
thank you.
Thank you, Vivek. Frederick, there are many people who want to meet you, but there is a person who I want you to meet. When it comes to data, when it comes to reliability of acquired data from systems, especially from OCT, but also from others, I have a go-to person. And that person is Giovanni Lucia from Aquiris. Thank you very much, Giovanni, for joining us. Could you tell us, and also, of course, tell Frederick in the hospital what you can do?
Hello, everyone. Of course, I can. Let me share my screen.
For those wondering, this is not a background where I am right now.
Can you see my presentation?
Crystal clear.
Yeah, we can see it.
Okay, so I work at Aquiris. I am the swap source solution manager. At Aquiris, we provide swap source signal acquisition technology, so data acquisition technology for different applications, but one of our main application is swap source. And we follow the customer from the concept to the production of their medical instrument, basically. And we provide an easy-to-use solution because it's a complete engine which is provided. So we have the entire processing chain on board, real-time FPGA, and this allows to get the best performance. Everything is really optimized to get the best quality image and with less artifacts. We have really superior image quality thanks to our specific technique for resampling, and we have independent from laser source, and we also can provide really enhanced resolution, for example, for surgical guidance and disease detection. Our technology can also complement other type of imaging technology. So today we see imaging or we see OCT added to some of the already existing medical equipment like surgical microscope, for example. We also simplified the system integration. We have integrated IOs, including galvo control, simplified synchronization, a modular design, and a full set of driver. So really, if you want to de-risk your technology, you need to acquire data, process data. We are the expert in that field, and swap source OCT is really where you need a highly competent company to help you integrating in your instrument and get the best performance.
Thank you very much, Giovanni. When it comes to data acquisition, where do you stand, Fred? Is there any challenges, current activities, current projects that you see maybe a mutual room for collaboration?
Yeah, I think that one of our challenge basically at some point becomes the size of the data sets that we have basically, and the cross-platform nature of our data sets from biofluid to surgery. So there's a bit of a challenge we are is making sure they're accessible and can be probed, data mined, simultaneously all together. So, and as the data sets are growing, we will get into issues in managing all this business. So this is the challenge that's coming, but we don't really have yet, basically.
Yeah, that's why we have FPGA processing on board. Yeah. Everything is done in real time to reduce the amount of data and pre-process the data so you can get basically your result. For example, for OCT, of course, you can stream raw data and do the processing on the host, but this, of course, requires a lot of processing, a powerful host, and you have a lot of data to manage. We can have the entire processing chain on board, meaning that you can read the B scans, for example, and plot your image directly. So basically, very small processing is required to show your image and build the medical instrument. The entire engine is in real time on board.
Yeah, I think
congratulations on everything that you are achieving, and I think it is great, and there are many people who are going to be reaching out to you because of what you just said. Frederick, back to you. There is more people who I want you to meet. One of them is the company Hamamatsu has now a pilot line for production of customized solutions. Andrei Voloshin, thank you very much for joining today from Hamamatsu pilot line. You are the chief technologist. Tell us what's on your mind.
Thank you so much. And this is really cool to hear and listen and see how the photonic technologies are going into the real world applications. So I'm happy that right now marketing people like the first speaker is pitching photonics for medical applications, because we know that photonics can really change the world, but we have to make it happen. So we have to think out of box how our solutions can really change it. And we have to partner with each other. For example, this OCT swept sources electronics is extremely important for OCT. And if you need some custom photonic modules, optoelectronic PCB from us, I'm happy to help. And if you need a contact in the US, I'm working in Europe, and from the US, we have a special dedicated team that can help you to build the module for application.
Awesome. Thank you.
Thank you.
Do we have Brent from Stryker still in the room?
Yes, sir. I'm here.
Brent, I don't know if you saw the data acquisition system presented by Giovanni from Aquaris. I would like to ask you, is there any particular challenges that you have on this particular respect on data acquisition?
That's a good question. I probably can't say for sure from a data acquisition perspective what the challenges are. Certainly, one of the things that is very, very important for us is, and maybe this is tangential, but latency in that acquisition and image processing.
Yeah, good.
In surgical applications, generally we're looking from glass to glass, less than 100 milliseconds. And when you run that through a chain, not just the processing of the system, but then you also have latency as it gets routed through the operating room, out in the wall, up and around, with various, processing attached to it. Surgeons are and rightfully so, extremely sensitive to any sort of lag or latency, because they're working on, just minuscule structures. So, from a data perspective, while whatever data is kind of captured and analyzed for subsequent usage is maybe not so much an issue, but for us, making sure that all of that processing is able to happen as close to real time as possible, because we have tough customers who are, even if I don't notice a lag, they'll pick up a lag for sure. Similar with sensitivity to any sort of strobing lights, even at a much higher rate of frequency than many of us would be able to detect. after using it for several hours, they'll complain that, hey, this is giving me a headache, etc. So that's probably a little bit tangential, but it is an important one.
I think it is an important point, and it is for us because, for example, for guided surgery, when OCT is used, you really need to have a very small delay from the moment the surgeon operates and he can see the effect on the image. And for doing that, for example, we can transfer data directly to the GPU for further processing without going to the host. This allows, for example, to have the fastest and shortest path for the data to get processed and then visualized by the surgeon. So this is an important feature, and you need an ecosystem and infrastructure to allow to integrate a data acquisition module with processing, a GPU, and everything combined to build a medical instrument. So we do have this infrastructure in Accuracy, this ecosystem.
Great point. Thanks, Giovanni.
Yeah, thank you so much. And this is one of the things. I mean, Aquatic Systems also works very well in endoscopy and other systems. So and the latency is actually very well controlled. So this is something that I would like you to say a little bit more about it, and if you can say a little bit more how you can help just in general for the robotic surgery.
Yeah, so in general, so we do acquire the data, and those data get routed to an FPGA. FPGA has a very controlled latency. When you implement something, you can actually measure exactly the time it takes to get the results. And once you get that, then those data needs to be sent to somewhere else for further processing. Now, this sent to somewhere else is quite critical because, for example, if you're reading data from an host via software, then somehow the latency is not predictable. And then you have some delay, and this is not really acceptable for doing guided surgery or when you have to drive a robot, for example. Then in this case, what we can do is that we can send directly from the FPGA with a known latency to a different location, for example, to a GPU, where also the latency of the processing is very well controlled. And once you have the results, these can be then displayed in the host for the surgeon, or provided to the robot as input for guided surgery, for example. So latency is somehow a factor to take in account. For some of the application, is not really mandatory, but for guided surgery or for robotic surgery, then when OCT is used, latency needs to be mastered.
Okay, thank you so much. So now that we are talking about OCT, I would like to call to the floor Alastair Price from Siloton. So this is a beautiful project, Alastair. Please tell us what is your project, what you need, what is your challenge, and what you can offer.
Yeah, absolutely. So hopefully you guys can see my slides now.
Yes.
Brilliant. So yeah, my name is Alastair Price. I'm the CEO of a company called Siloton that's based in Bristol in the UK. We develop photonic chips for OCT, and there's a wide range of areas this can be applied. One or two have already been mentioned during the meeting, but the area that we're beginning in is eye disease. This is the area that OCT is mostly used at the moment. It's a big problem. About 420 million people have eye disease, and just to put that into context, so the number of people who have been diagnosed with cancer over the past 5 years, it's about 7 times the size of that number of people. It's a really big problem. We just don't often hear about it as much because it doesn't directly kill you. But it does have some quite serious impacts on people's lives. And right now, people are suffering from fairly inefficient care pathways. They can go into a hospital and get an OCT scan of their eye to see what's going on, identify what disease they have. However, if that's a disease that needs regular treatment, they can't then receive that regular monitoring OCT scan anywhere other than in a specialist setting like a hospital or an optometrist. Now, this means that patients' lives are disrupted quite significantly because they have to make very frequent trips to the clinic. In some cases, if the patient lives in a remote area, this can, be an hour and a half they have to travel. But at the same time, then, 1 to 6 months is the interval that you'd normally go for between bringing people in for another scan, see if they need to be treated. That is too big a gap for caregivers, so the doctor, to catch any unexpected changes in some of these conditions. And so really the solution at the moment is just to massively overtreat patients to reduce the risk of unexpected sight loss, although it doesn't eliminate that at all. And the key thing is these diseases cannot be treated without an OCT. You need to have that scan to know exactly what's going on in the eye to inform that treatment. So really we just need to make sure that OCT is more accessible. We need to bring it out into communities so that people can scan in the home if they need to do it a few times a week, or in community centres if they can do it maybe once every couple of weeks. That'll mean that we can pick up any unexpected changes a lot faster without disrupting lives. And there are some health economics that have projected a saving of about £1 billion a year for healthcare providers like the NHS. And the way we do that, this slide is our kind of general slide for people who don't know what chips are, so I won't spend too much time on it. But yeah, photonic chips, as I've mentioned. That chip there is our first generation AKEPA device, artistically illustrated to avoid any IP leakage, but that's our first generation chip there. And as I mentioned, eye disease is the first area that we're looking at, so ophthalmology. Beyond what we just discussed there are applications for pharmaceutical trials as well. So when you're developing new drugs or new ways of administering drugs, being able to regularly monitor people for things like intraocular inflammation, which can be a side effect of those drugs or those methods of delivering the drugs, and which itself can send people blind. It's really, really important to have regular OCT monitoring for things like that. However, there are other areas as well, particularly otology, so looking for ear infections. Dentistry and dermatology are other areas too, where the same kind of chips that we're developing here could be applied. And then in terms of what we're doing, so we became the first people to image patients' eyes with photonic.
It just I think it got
This is
frozen.
the beauty of doing the meeting live. You go to Bristol and then the signal gets in the middle of the suspension bridge, and we have lost connection with Alistair. So let's wait for Alistair to slowly come back. And in the meantime, there is a lot of people in the room who are asking questions and looking for connections. Why don't we go to one of the key companies in the field of adaptive optics? I am really happy to have Stefan Weber with us from the company Faceform, all the way from Berlin. Stefan, are you with us?
Yes, no, thank you, Jose.
No, thank you and congratulations on everything that you are achieving with Faceform. You were one of the first companies to join Optica when Optica created this new way of understanding industry, and I can see that 3 years after, things really are going your way. Stefan, the floor and the attention of everyone is yours. Tell us shortly what Faceform is and how we can use it.
Definitely. So Faceform, so we are, well, a startup, not yet a scale-up, but Faceform is now nearing its 5 years anniversary. And we have been doing adaptive optics in a very different way than it's been done before. You would think, how different can it be? But, there's in optics, there are always two different ways how light can interact. It can be reflected, or it can be transmitted. And actually, we're the ones who bet on the transmissive part. So like a window and a mirror, right, has very different functions. Even light interacts in a same way, in some way. So what you can see here is our technology called deformable faceplate. So it's essentially a piece of glass that has a little bit of liquid encapsulated, and then there is a membrane that presents the boundary, and we apply high voltages from inside the aperture, and therefore we can create like a reconfigurable freeform optical surface, adaptive optics, thin piece of glass that you can essentially put anywhere. So it doesn't reflect the light, it transmits the light. It can do high order Zernike's up to 8 order. And it's a general purpose wavefront modulator as well. Because our previous speaker was talking about OCT, maybe I'll start with that particular example and also talking about miniaturization and retrofits. So you can actually put this device into any, well, optics device, for example, an OCT, as we showed here with a customer in Paris. And they added this to their miniaturized OCT, and eventually it corrected for all the residual eye aberrations, and that gives you cellular level resolution. So this can do up to 2-3 microns in resolution, something it's very hard or even impossible to do without adaptive optics. So we can also do improvements in microscopy, particularly if you go deep into the tissue. There is another example where you can actually just put the device after a microscope objective, and then you can see, for example, here the neurons or the dendritic spines much more precisely. What we also do is now adaptive optics modules. So if you can't actually slice it anywhere in your device, you can attach it in front of any commercial microscope. And we also have software now to optimize on an image. Also towards our optics studio, ANSYS CMAX sponsors. So we have also empirical ZMAX model. That means you can put that software component, the DLL, just in your device, and you can see how much you can improve your device in principle, and then you can decide how you wanna and where you wanna implement it. So just a few words from Faceform. I wanted to stress out transmissive makes the use of adaptive optics very easy. You can put it anywhere and see how you can improve your image.
Thank you.
Congratulations again. Go ahead.
Yeah, many thanks. Congratulations again, exactly for what you are achieving now. So I have one question for you, and it's like when you are using your adaptive optics in deep tissue, different techniques. Like, do you not only get more better image, but my question is about like how deep you usually can go in the improvement.
Yeah, this so there is of course the 2-photon microscopy, even 3-photon microscopy on the horizon, and there adaptive optics is even more critical, and there you can go really deep, like millimeters. So with 2-photon, it's hundreds of microns. 3-photon could extend even further, and there's all kinds of distortions inside the tissue that you cannot predict, or even the most expensive microscope cannot pre-correct if you don't have any adaptive optics.
Okay, so that's good. So we have Andre again asking a very good question. So Andre, can you please open the mic and say it yourself?
Yeah, so we have in Switzerland an Optotune technology, which looks very similar. I want to know the difference. And could you make a 2D phase image?
So Optotune is essentially a 1 degree of freedom version of what we have. So it controls the focus. Does that very well, does it to a great extent. So we start with actually 63 actuators. So we have 63 degrees of freedom to start with. It can do spherical correction, trefoil, and combinations of Cernicus. So it's extremely versatile. And Optotune, it gives you an extremely broad and broad focus. And you can, of course, combine them, which we also have been doing, because they are all transmissive elements. You can use them in sequence. And for the phase imaging, it's essentially a similar question. Of course, if you want to just do a phase offset, like that's maybe not a bit of an overkill. But if you want to do phase imaging with some, let's say, continuity on an optical surface that doesn't get you any additional diffraction effects or changing the polarization, that would be actually a very good idea to try.
Yeah. So also, I would like to go back to Frederick, because Frederick, this is the kind of optics that it could be interesting for you. So do you have any comments on this?
Yeah, well, this is our challenges basically are always improving do photonic sensitivity, basically. So anything that can be used to do that, we're willing to test, and work with. So yeah.
Yeah, so hopefully we can put you together. So yeah, Stefan.
To try.
We
Yeah,
are willing to pay for the traveling and for the shipping cost of the optics of Faceform from Berlin to Montreal. It's included in the membership fee. Helena, I think the meeting is going extremely well. I think the companies are really sharing their challenges, I would like to invite everyone in the chat so they can write some of the challenges, a room for collaboration that they already have observed over the next hour of the meeting. Helena, what's next?
So meanwhile, before we go next, so I would like to call Matthew Bergon from Excel because he also has a question. So we were talking before about the optical fiber. So and this is something that somebody can help here. So Matthew, can you open your mic and your camera? Thank you. Good to see you.
Yeah, sure. Good morning, good afternoon, everyone. Yeah, I was just wondering, so you have this optical source, maybe one or multiple lasers, you're sending the light to the tissue, so to the probe. We haven't discussed much about the optical fiber in between. Is there anything that could be improved on this side to improve the overall performance of the system? So I think you have maybe two fibers, one to send the signal and maybe one or multiple fibers to collect the backscattered signal. Thank you very much.
So Brent, I think this is for you.
Sorry, say again about the optical fibers. I didn't quite get the second part of the question.
Yeah, so I'm assuming you are using optical fibers to send
Yes,
the
sir.
signal from the source to the probe. Is there anything on this side that could be improved? Maybe better transmission, broader transmission window to accommodate more wavelengths, maybe?
So yeah, to kind of help the group understand the way that most of the surgical camera setup is none of the data is transmitted over the optical fibers. That's only the illumination that comes in. And in that case, generally speaking, the biggest complaint that we hear is not a particularly challenging or technical one, but it's one of practicality, is that these devices are all used in the sterile field. And certainly in North America, they are sterilized. Sometimes in Europe, often in Europe, they're draped. But they're sterilized, which means they receive a lot of heavy cleaning and reprocessing in high temperatures, like, autoclaves, etc. So one of the complaints a lot of times with the fiber optic channel is less around the transmission and more actually the durability of the fibers themselves. Because, you can imagine if you're doing 2 or 3 cases a day, it's getting turned over, it's being moved downstairs for reprocessing, you have a lot of broken fibers. And those broken fibers over time obviously lead to less transmission. So the complaint that we typically hear on the fiber optic side is twofold. One is it's too thick or too heavy, especially for pediatric patients. They say, hey, I want a smaller form factor. The other is, I've used this for 6 months and it ends up being, I'm losing too much light, and now as a result, I'm, having to turn up the illumination of the source because I'm losing something. Now, the flip side is, be more careful, but it kind of goes back to, and in the handling of your equipment, but it goes back to that comment about the workflow where, we try to accommodate what is existing in the hospital. And if they want to beat this stuff up, it's kind of incumbent on us to be more delicate. So I think that for me, it's less, the challenges we see for fiber optic cables is less around, the exact parameters of transmission, because we're not sending data, and much more around, okay, the size of the bundle and also the durability of the bundle.
Thank you.
Yeah, of course.
Thanks a lot. So one of the other things that we were talking before, and Stefan mentioned, is about how to do all the simulation and all the things. And we actually have here Sia from ANSYS. So Sia, can you tell us a little bit what are the capabilities that ANSYS can do for medical imaging?
Yeah, hello all. Let me quickly share my screen. Yeah, can you see the screen all right?
Yes.
Okay, yeah, so welcome everyone. My name is Csilla Timár-Fülep and I'm the application engineering team lead for the optics and photonics workflows here at ANSYS. So the ANSYS Optics portfolio enables multiphysics and multiscale simulations going from the nanoscale, starting with ANSYS Lumerical, which enables us to do photonic design on the nanoscale level, on the chip level. Then ranging to the macro scale, we have ZMAX Optics Studio to design optical components. That means design, optimization, and tolerancing of the lenses. And up to the system level where we have ANSYS PIOS to simulate, visualize, and validate all the simulations in a full 3D environment, taking into account how a human observer or a machine vision system would see the images. Using all these tools, we of course have solutions for the medical industry as well. Just to name a few, I have listed here some applications. Some of these have been already mentioned in the meeting. So we can simulate microscopes, whether it's phase contrast, confocal, or fluorescent imaging. We can design spectrometers for wavelength-dependent material analysis to investigate biological tissues. We can also track the phase of the light, so we can easily simulate OCT systems to investigate 3D samples, for example, the human eye, as mentioned before. To help non-invasive imaging technologies, we can design endoscopes to look inside the human body or design micro-objectives to facilitate robotic surgeries. On the bigger scale size, we can design surgical lighting to offer optimal illumination in the surgical areas. And our simulation tools are not limited to the optical wavelength. We can go to the infrared regime, so we can design thermal detectors, for example, for non-contact body temperature measurement. Or at the other side of the wavelength scale, we can simulate UV or even X-rays. That means we can model X-ray medical imaging to investigate the internal organs and their structures. And with all these simulations, we help research and development to achieve a better performance, a better reliability for all the devices, and replace physical prototypes by virtual prototyping and help to get those devices faster to the market. So if any of you are interested in more details on how we perform these simulations, what are the details, please feel free to contact me and I will be happy to share more details around it. Thank you.
Thank you so much, Silja. Thank you so much. So now that you were talking about microscopy, there is a company that is called Copper Surgical Fertility Solutions. And basically, this is going to be one of the leaders in reproductive technology, in vitro fertilization, and how could involve all the system for OCT and embryo selection. And it's my pleasure to introduce you to Austin Lindley from Copper Surgical. Austin, the floor and attention is yours.
Hi guys, thanks very much for the introduction. We're probably going to talk about something slightly different. Let me get my screen up. Share. There we go. I think what we're going to discuss is how the equipment that we supply is used in fertility, in particular the Integra 3. So my name's Austin Lindley. I work for Cooper Surgical and I'm the regional manager for fertility sales in the UK and Ireland. Let's hope this works. Here we go. Okay, so I'm just going to give you a quick company overview because many of you probably have no knowledge of Cooper Surgical. And then I'm going to talk about our micro manipulator, which is the Integra 3. I'm going to give you a product introduction, a bit of technology, look at the applications, the support, and just give you a bit of a summary at the end. Just as a company, Cooper Surgical has a global representation, predominantly direct, but we do have some channel partners. if anyone has this statement here, every 30 seconds a baby is born using Cooper Surgical's product is true. If you have been through fertility treatment, you will, or at some point within that treatment, your journey would have been or would have involved a product that has been designed, developed by Cooper Surgical, either equipment or reagents. We are the largest global supplier of products and services to the fertility market, and that just sort of exemplifies the impact that we have in the field. And as a UK organisation, we have a direct sales and service representation, 7 sales, 6 service, and we have our own customer service in the UK. And the whole of EMEA organisation is pretty much direct with sales and service. So just to give you some introduction, let me just bring this video up. And I need to slow it down because it rattles through quite quickly. So the Integra 3 micromanipulator is the most recognized micromanipulator system in fertility, combining, and here is the instrument here, combines the manipulators, the XY stage, and the heating elements all in one design. The heated area incorporates a product called ThermoSafe, which is unique to Integra 3 and bathes the area in warm air, so you don't get any cold spots, so the objective doesn't draw any heat. There's also a very large heated area, which is controlled independently. So you've got 3 heated areas, the ThermoSafe and the 2 heated plates. We have a manipulator, which is a fast pipette setup. And exchange. The mechanical unit itself is completely mechanical and has 5 positions of control being displayed here to give you flexibility. We have a smooth course control and movement of the manipulator. And it's ergonomically located within the XY control for the stage. On the left-hand side, we have a status indicator. So if any of the heated elements go out of range, you'll get a visual and an alarm to determine that. And we can also, you would have seen many of these images on TV. We can create movies, take pictures directly from the system. And a lot of people like the fact that we can use as a counter, so you've got a counter positive and negative, and also there's a stopwatch so you can time your protocols or whatever you're doing. Under the stage, so we fit this onto a microscope. Under the stage, it's very nice actually, because sometimes people want to see what's going on underneath, what objective they're using. So we have understage lighting, which is motion sensor, and the unit will connect to the main brands. Olympus, Zeiss, Leica, and Nikon. So just to touch upon some of the technology, we mentioned ThermoSafe, and this gives you a comparison between ThermoSafe on the left, a standard heated stage without any ThermoSafe, and you can see that the objective creates a cold spot in the middle. And a glass ITO, which is better, but it does still give you a sort of cold spot in the middle. And just to give you a look at what ThermoSafe is, so you have your objective and you have a nozzle which just bathes that area with warm air around the heated metal insert. We have flexibility, so we have our own syringes. So we have a coarse and a fine syringe, and we also produce our own oil syringe, but we are compatible with Narashigi, Eppendorf, and also the Eppendorf Femtojet. The manipulator itself is totally manual, and it has no electronic components or pneumatic components. And this, as a product, provides good feedback to the user, and can be adjusted to suit that individual regarding the tension on the movement. Some of the applications, so this is the type of thing that you would see. It's an egg that's being injected. You've got the holding and the injection on the right, the holding on the left, the injection on the right. And the orientation is correct because if you see there's a small polar body, so a small cellular mass at the top. Hopefully over time, so 4 to 5 to 6 days, you'll start to see an embryo develop, and this is a multicellular phase. And in the end, we hope that this goes on to become a transferable embryo and a successful pregnancy. But that's the staple of our work, but a lot of people don't know we also deal a bit with canine. And we do a little work with canine at the veterinary colleges. We do some bovine, and one of our largest secondary markets is equine, and the equine ICSI is quite a large business globally. The company itself is ISO registered, and all our products are MDR, and we have our own service facilities where we can support these products, the installation and the ongoing support. So just to give you a summary, the product is unique. There is no other device like this in the market. Compatible with your existing hardware if you have one of the main microscope brands, or we can supply a microscope or whatever. Trusted in the market, so it's the device of choice for fertility at this time. Flexible, so specifications, we can adjust the specifications to meet your demands and adjust sensitivity. And as a product goes, it's established in the market. This is the 3rd generation of this system, and it is the market leader. Thank you.
Thank you so much, Austin. So this is actually really nice. So that is one of the typical questions that I get when we talk about this kind of devices, and is what are the optical requirements? What are the optical requirements that are really critical for safe and effective embryo laser surgery?
So we use I haven't spoken about a laser, so we do use a laser, and we produce our own objective for that product. It's an infrared laser which is used to ablate the zona to allow so that's one of the examples to allow the injection pipette to enter without actually punching through. There are other examples where they use it to collapse an embryo when they want to freeze it. And also if they want to do genetic testing, so we can actually punch a hole through the zona, remove small amount of the genetic material, and then cut that with the laser and then take that away for genetic testing. The microscopes themselves are pretty standard. They're an inverted microscope, and they're all fitted with Hoffman. Everything is in plastic. There's no glass interface, so we don't use DIC, and there's no fluorescence in the clinics, although there's probably fluorescence going on in research institutes. Does that sort of answer that question for you?
Yeah, sort of. And yeah, also this is like but the ideal is like because your laser is a Gaussian profile, so how good have to be the profile? Let me put it that way.
The action of firing into a zona needs to be very precise. Obviously, you know the actual to put a hole into it can't be damaging. So I think Brent, you spoke about damage to cells from overexposure, and I think that that's something that's brought up quite a lot. We have I can't remember the stats for the laser, but we have a higher power laser, which means that it's a shorter frequency to generate the hole that's required, whereas others go for a lower power laser with longer frequency.
Yeah, so I would like to call to the floor to Jax from Fabulem's Optics, because Jax has a little bit for everybody here, to tell the truth. So Jax, the floor and the attention is yours. Please tell us about Fabulem's Optics, what you do, what you can do for others, and what others can do for you. Give me the Optica question.
Very much. So I'm from Fabuland and the sister company MCD, Modern Camera Designs, and I will take you back to the very basics of imaging or image acquisition, which means lenses and objective lenses. Probably many of you know compact camera modules made from waveform optics from OmniVision, and those are very often used for single-use endoscopy. But there are problems in scaling the image quality for larger images or higher resolution images, and also some supply chain issues. We have taken the UV replication approach one step further from waveform optics, trying to combine it with the advantages of optical injection molding. So we, as a startup, are developing a waferless and shrinkage compensated UV replication that can give the image quality and degrees of freedom of the lens shapes as injection molding, but an adaptable degree of batch parallelization as, for instance, waveform optics, so that many lenses can be manufactured in parallel and provide very cost competitive lenses and objective lenses. At the same time, the materials we use are high temperature polymers, also known from waveform optics, so they can, for instance, for single-use camera modules, be reflow soldered to PCBs, or also withstand autoclavation temperatures and many cycles for reusable or multi-use endoscopes. The molds we use only have to withstand room temperatures, so the costs for those, and with that the NREs for new projects are quite small. So we have done quite a lot of systems already. Typical parameters you can see, for instance, on the lower left, an endoscope with 120-degree field of view, typical image sensor. Here we have different materials for acclimatization to achieve a good image quality, and all of this fits into a 2mm diameter barrel. We are also seeing trends to provide single-use endoscopy for 4K images, and we are doing developments in that direction as well, in order to provide higher resolution and image formats. So if you are not happy with your current lens supplier, either for cost reasons, design degrees of freedom reasons, temperature stability reasons, we are here to give you all the benefits of polymer replication process that can provide similar stability and quality as glass lenses. So get in touch with us.
Thank you so much. So I mean, there is a lot of things that you can do for Copper Surgical here, and also for Stryker. And I would like to see all the collaboration also with the Shun Hospital, not only with Frederick, but other companies that they are running out of over there. And I would like to call the last speaker for today. So is Mr. Tom Chen from Flex Compute, and he's going to talk about the tidy 3D platforms and an electromagnetic stimulation that is very useful for photonics. Tom, the floor and attention is yours.
Great, thank you very much. Let me get my screen share. Yeah, hello guys. My name is Tom Chen. I'm from Flex Compute. Our company specializes in GPU accelerated, ultra fast, and accurate physical simulation software. Today we have a lot of speakers mainly from the hardware side, but let's say software layer that really powers the design of all this amazing hardware. So we help engineers and researchers to model and design very complex optical systems with unprecedented speed. Since today's event is focusing on optical imaging and visualization in medicine, that's where imaging quality and sensor performance are critical. The example that I'm showing here really demonstrates how kind of full wave electromagnetic stimulation can be applied to optimize a CMOS RGB imaging sensor. They are commonly used in many of the imaging systems. So with our native geometry engine, we can very rapidly construct the full pixel stack geometry, including all these micro lenses, color filters, metal layers, and photodetector layers. And the versatile dispersive material models really captures the behavior of color filters. And the broadband time domain simulation allows us to simulate the broadband response, which is super critical in just a single simulation, which is cannot be done in the traditional frequency domain-based methods. So on the right, you can see how the red, green, and blue light focus differently at the detector layer. And below is the simulation result for the optical efficiency. This type of detailed modeling really supports the design of high efficiency and very low noise sensors, and ultimately enabling clearer and more reliable optical imaging. If you are working on imaging systems and would like to see how advanced simulations can help and accelerate your design, we would love to talk to you and learn more about your applications. Yeah, thank you very much.
Thank you very much, and thank you all the great speakers that we had so far. What we're gonna do now, for everyone in the room, for all our friends, for Brent Summer, for Stefan Weber, for Antonio Raspa, we are gonna close the live stream. If you want to stay for a quick discussion afterwards and have a virtual drink with all of us, it is great to have you. But for those of you who joined the live stream meeting, well, that may have been a bit too loud. I would like to ask my IT department to quit now the live stream.